All right, so thanks everybody for joining me for the science chat on July 1st, 2020. My name is Ian Breckheimer, and I'm a staff scientist at Rocky Mountain Biological Lab. I'm actually pretty new to the job. I started in January, and my background is as a landscape ecologist. And what a landscape ecologist does is basically make maps of nature, different aspects of how the natural world works. And we then use those maps to try and figure out how different parts of nature are connected together. And so I'm really excited to talk about some of the projects that I'm working on and some of the tools that I'm using that generate really amazing maps of the places around Rocky Mountain Biological Lab. And so this is going to go, I'm going to talk through a few slides and then actually show you guys around some maps because some of the stuff is better to show than to tell. And then there are 3 points sort of along this when I want to stop and open things up for questions or for discussion, and then we'll move on. But first of all, thank you so much for coming and joining me. I'm excited to talk about my work. I'm going to go ahead and share my screen. I have a PowerPoint presentation that mostly is just photos, but I will get that up right now. Alrighty. Okay, so you guys should be able to share, see my screen. Please, if you can't, please stop me. Okay, so by now we've like become kind of used to seeing pictures like this. This is this is 6 different views of the Earth from space that are taken from different viewpoints. You can see sort of the continent of Africa on the top left, North America on the top center. This almost entirely blue one on the top right is the Pacific Ocean. That's like it's amazing how giant the Pacific is. It takes up almost half of the globe, really. And of course, down here, the lower right, this is what the Earth looks like from space at night. So you can see the little blobs of yellow light are major cities. So images like this have really been a revolution for scientific understanding of the environment. And for the first time in really this is like in my parents' lifetime, we started to see this bigger picture of how the world works. We can see how clouds are affected by the ocean. We can see how the seasons change the color of the Earth's surface as it greens up and browns down with the seasons. And we can start to understand the connections between the Earth and the atmosphere in ways that that really allow us to understand important things about our climate and how we're impacting the environment. And images like this come from very sophisticated, essentially telescopes in space. And I just wanted to show you what one of those looks like. This is a satellite called Sentinel-2, and it's launched about, I think 4 years ago by the European Space Agency. And this might not be super obvious. This is just a giant telescope. So the lens or the mirror opening is sort of facing down, and it's in the process of being packaged up into the top of the rocket that gets shot into space. And then it unfurls its solar panels and turns the telescope towards the Earth, and starts taking pictures. This is a really fantastically sophisticated instrument. It took about 25 years of planning and engineering, about $800 million of funding. And what it does is it circles the Earth and takes pictures. And those pictures essentially look like this. So this is a picture from about a week ago from this satellite of the Rocky Mountain Biological Lab campus in Gothic. And one of the things you'll see is that this image is kind of blurry. And it's not because there's something wrong with the satellite. It's just that it's trying to take take pictures of something a couple of 100 miles away. And so there's atmosphere in between, and it's just really, really small from the perspective of the satellite. And for the most part, for the past 4 or 5 decades, this has been the view that we've had of the Earth. It's been a little bit blurry, and of course, there are satellites that can take sharper pictures than this. For example, spy satellites that are run by the National Security Agency, or NSA. Those pictures are considerably sharper, but that's a telescope that only takes a picture of a tiny little sliver of the Earth, and you have to sort of figure out where to point it. So if you are worried about missiles in North Korea, you can tell it, point it at this particular spot in North Korea, and it can take a sharper picture. But this is one of the satellites that just takes pictures continuously. So it passes over every point on Earth about every 5 days and takes an image like this. So these are really useful for tracking larger scale processes. So if we were interested in snow, if we were interested in how forests green up and brown down with the seasons, this is fantastic data. But most of my work actually uses sensors that are trying to push this down to get sharper pictures. And the reason why that's important is that biologists on the ground are taking very fine scale measurements of plant growth and development, of the fate of individual animals, individual plants. And for the most part, this data is at a much finer scale than the kind of blurry coarse scale pictures we get from satellites. So if we want those two data sets to talk to each other, if we want to be able to use the field measurements and the remote sensing measurements, we need some way to get remote sensing measurements that are a little bit clearer than satellite imagery. So today I'm going to talk about 3 relatively new remote sensing data sources that we can use to tell us really cool stuff about how the world works. And I'm going to just give you a really high-level overview of the stuff that we're working on at Rocky Mountain Biological Lab using these new sources of data. So really briefly, I'm going to talk about LiDAR, which is a laser scanning technique that generates really high-resolution images of topography and vegetation. I'm going to talk about imaging spectroscopy, which is a technique that allows a really fancy camera to see billions of more colors than we can see. And then finally, I'm going to talk a little bit about drone imagery, the way that we're putting sensors now on platforms that are much closer to the Earth's surface than a satellite or an airplane. And that opens up a lot of new possibilities. So I'm just going to get started with LiDAR. So you guys have all used a laser pointer or seen a laser pointer. That's a little handheld laser that is usually red, and it makes a little dot on the whatever you're pointing at, or maybe you're using it to entertain your cat. So if you were to put that laser pointer on an airplane and then had it scan back and forth so that the laser was essentially like you're entertaining your cat, it just goes back and forth and back and forth and back and forth several times a second. And then there's a detector that detects the reflections of that laser off of the Earth's surface, so that the LiDAR pulse goes down, a laser pulse goes down, and then it records the returned energy from that laser pulse. And because different objects are different distances between the airplane and the ground surface, it can use the amount of time it takes for light to get from the surface back to the detector to tell how far away a given surface is from the airplane. And the airplane has a really accurate GPS on it, so it knows exactly where it is in space. And so we can use that to generate data sets like this. This is what's called a point cloud. And it's essentially take one of those laser scan lines, and this is all of the returns that the sensor detected. And they're color-coded by how far away from the airplane it was. So you can sort of imagine the airplane flying a couple thousand feet above this area of the Earth. And it returns a cloud of 3-dimensional points. And then we can process that cloud of 3-dimensional points to tell us something about the elevation of the ground surface. That's the sort of lowest elevation where you've got returns. And also something about the elevation of the forest canopy or the vegetation canopy. And so those two pieces of information are really basic things you can get from LiDAR, and they're just incredibly powerful. What it allows us to do is map terrain at really, really high resolution. So I'm going to get out of the presentation. I want to actually show you what these maps look like. And I'm going to show you what a LiDAR map of the ground elevation, what a LiDAR map of the canopy elevation looks like for Crested Butte. So I'm going to zoom in here on Crested Butte, Colorado. And what is shown on this map is what's called a hillshade. And that's a way of showing topography that sort of imagines the shadows from the sun coming from a particular direction. In this case, I think it's a southwesterly direction. So this is like what the Earth would look like, or what the shadows would look like in the late afternoon in the springtime. And so what you can see here is these are actually all the buildings in Crested Butte. So if you zoom in on Elk Avenue, it's going to get a little bit pixelated, but all these squares here are the individual buildings, and you can tell how tall the buildings are from this image. And so we can say more about this than just the buildings. Oop. I think I might be, oh, yeah, there we go. Had a little bit of network hiccup there. So it really gives us a really fine scale picture of what the topography looks like. And the things that I think are fascinating about images like this is they tell us a lot about the history of the landscape here. So one of the things you'll find is these kind of parallel ridges that are coming down from the top of the map and going through that right along the road here, Gothic Road. You can see those parallel ridges. There's some more down here. This is like the club at Crested Butte. There's all these parallel ridges. So these are leftovers from glaciers that used to be in this valley during the last ice age. So you can sort of imagine a glacier coming down the Slate River Valley and bulldozing a bunch of sediment and rock in front of it. And then when the climate warms up or it doesn't get as snowy, then that glacier then recedes back up towards the top of the valley, and it leaves behind these big features that are called moraines. So these are a set of parallel moraines, which are geologic features that were laid down pretty recently, so probably about 16,000 years ago, as that glacier receded up the Slate River Valley. So that's just one example of how we can use this LiDAR technique to tell us something about the history of the landscape where we're living. I also wanted to point out that it can show you about show you a lot of stuff about vegetation. So this kind of marbled surface on the hillshade, these are actually individual trees that are showing up. And you can process this image to actually get a pretty good map of vegetation canopy height. So this is the height of the forest canopy. So the darker green colors represent taller trees. And it tells us a lot about how recently this forest here has been disturbed. So if you zoom in on this map, you'll see some parallel stripes here in the forest that have lighter colors, indicating that the trees are shorter there. Now, these are avalanche tracks. So the reason why the trees are shorter there was that at some point in the recent past, an avalanche has come and totally wiped out the forest. I think that's pretty cool. Okay, so that's like, I guess, cool stuff that we can show you about LiDAR. I want to talk to you a little bit about the stuff that we're doing next with this data. So I'm working with Western professor Jessica Young on a really cool project to use LiDAR to map habitat for this creature here. So this is Canadian lynx. They're native to this part of Colorado. They're here. They're pretty rare, and they're so elusive. You would be lucky to see one in your entire life in the Gunnison Valley. But one of the reasons why you don't see them very much is they're really active in the winter, and they absolutely love snow. They have a couple of adaptations that make them really good hunters of small mammals in snowy environments. And they also really like dense understory environments. So that allows them lots of places to hide and makes them much more effective hunters. So if we go back to this LiDAR point cloud, what I'm doing with Jessica is actually trying to figure out if we can map understory cover, understory density from this, this LiDAR data, essentially trying to figure out what's the density of kind of blue and green points that might be aspects of the forest that matter to lynx. And we're just at the start of this project, and we've got a graduate student who's out collecting field data this summer to help us out with this. But I'm really, really excited that we're going to be able to tell really across the whole landscape, how good is this patch of forest for lynx? And that could help inform management. So the Forest Service makes a lot of decisions about how to regulate timber sales within forests like this. And what we want to do is identify those areas that are really important lynx habitat that should be conserved and maybe protected from timber sale. So that's what I'm doing with LiDAR. I wanted to pause really fast and see if you guys have any questions about LiDAR, how we're using it, or if there are any other things that that we can like look at and explore. If you guys are interested to see like you know, what landslides or avalanches look like in LiDAR data, it'd be fun to do a little bit of exploring. But I just wanted to give it up to you guys to see if there are any questions at this point.
I'm not seeing any questions in the chat right now, Ian.
No worries. Well, I'm happy to take stuff at the end, or and please, please interrupt me if folks have questions. I can kind of get on a roll. Okay. All right. So that was LiDAR. Let's talk about imaging spectroscopy. So most of you guys have probably been introduced to this idea of the electromagnetic spectrum. The idea being that our eyes can see a certain type of light that has particular properties. In particular, it's got a pretty narrow range of wavelengths. So the visible part of the spectrum has wavelengths of between about 400 and 700 nanometers. Nanometers are a very small unit of length. It's something like a billionth of a meter. And the reason why we're able to distinguish objects in the world by their color is because they reflect different amounts of radiation at different wavelengths. So if I look at a tree, what I'm really seeing is that tree is reflecting a lot of green light, and it's absorbing a lot of blue and a lot of red light. And that's also true outside of the range of the spectrum that we can see with our eyes. And sometimes those differences between different substances, for example, water and green vegetation, are much larger in the infrared part of the spectrum than they are in the visible part of the spectrum. Now, cameras take pictures in different ranges of this spectrum. And an imaging spectrometer is a really special camera that takes pictures in lots and lots of different positions along this, this electromagnetic spectrum, from the visible all the way into the near infrared. And what that gives you is a very special photograph. So a regular photograph you can think of as having kind of 3 layers. They're made up by pixels of red, pixels of green, and pixels of blue. And those are kind of overlaid on top of each other so that we can see it on a computer screen or on a printout. So this type of camera, instead of having 3 different layers, it has about 420 different layers. And each of those layers represents a different position along the electromagnetic spectrum. So some of them cover the range of radiation that we can see, but most of them cover this infrared part of the spectrum. And what that allows us to do is pick out the spectral signature of particular types of materials like water or roofing or soil or vegetation. It also allows us to pick up on pretty subtle differences between different types of vegetation. And what that's allowing us to do is to make maps of all these different kinds of surfaces at really high resolution. This is based on data that was collected in 2018. And I wanted to show you what that's allowed us to do. So this is, let me get off of the canopy map here, and I'll show you the land cover map. Okay, so this is a 1 meter land cover map that just categorizes the land surface into about 8 different categories. So the buildings here are in red, the conifer forest is in dark green, the aspen forest is in light green, and all the meadow vegetation is in this kind of yellow color. Oh, and the water is, of course, water is blue. So what we've done is to make, using this imaging spectrometry data, we've been able to make a really high resolution land cover map that covers the entire upper Gunnison Basin. And that's really useful for, say for Jessica, who's interested in lynx. Lynx really specialize in conifer forest habitat. So now we have a map of where all the conifer forests are in the entire basin, or sorry, the entire upper basin. But we're not actually done with this yet. Our next task is to try and make an even more nuanced vegetation map that tries to identify individual species of trees, shrubs, and herbs. And we're not there yet, but we're getting there. I wanted to show you kind of what we're going for. This is essentially a map of just a kind of a mess of colors. But what these colors represent is different types of vegetation. So I'm going to zoom in here on this area around the lower Slate River, right north of Crested Butte. So you can see very different colors up here on the ridge where we've got a little bit of aspen, we've got some dry meadow with sagebrush and lupin, and down here on the lower left side, we've got a bunch of different colors that represent different kinds of wetland vegetation. So we don't know exactly what all these colors mean yet, and that's a big part of our work right now, is trying to match up clusters of pixels that all have similar spectra to what does it actually mean on the ground? What does a lupin pixel look like? What does a sagebrush pixel look like? And that's going to take a lot of field work and quite a bit more kind of manipulating the data, but that's what we're going. We're really trying to identify every tree and shrub in the upper basin to species, or at least to genus. And that's going to be a boon to researchers who are trying to find different species. Say, if a researcher wanted to study lupin, they wanted to establish research plots, they could use this map to find really dense areas of lupin to suit their research purposes. And we're really just scratching the surface on what we're able to do with this imaging spectroscopy data. You can also use it to measure important things about the chemistry of leaves. And we can also do fun things like measure drought stress in trees using this data. So we're really just at the beginning of what we can accomplish here. All right, I'm gonna stop and ask for questions about imaging spectroscopy. If anybody's got a burning question about what we're doing with this very special camera, this would be a good time.
Hey Ian, it's Micah Russell. I have a question about this. Can you hear me?
Yeah, thanks, Micah.
So really cool that you're trying to get to species level classification. Mm-hmm. That's going to be a ton of work. Have you started working on that, spectroscopy library already?
Yeah, so we have about 700 field samples of essentially pure pixels of particular species. And part of that was done in 2018 during the initial field campaign for the Airborne Observing Platform acquisition. Mm-hmm. And then we've continued to do that for the last 2 years. So the library is growing.
Great.
That being said, we have a lot of samples for the common stuff, and we have relatively few samples for the rare stuff. So a big challenge ahead of us right now is trying to figure out how to deal with relatively uncommon plants and and in particular, like just as an example, almost all of the deciduous trees in the basin are aspen, Populus tremuloides. But there are a few black cottonwoods. So those two trees are actually pretty closely related, and they have similar leaves. But we're not sure if we can reliably distinguish between aspen and cottonwood right now, and that's a big challenge. Is we actually we need more cottonwood samples. We need to go out into the nature and find some cottonwood to sample. And that's just an example of one of those problems that we actually find that in a lot of different contexts where we'll have a really common species of lupin, and then we'll have like 3 or 4 that are really uncommon. And so what I can say is we're probably going to be able to do a good job for the common stuff, but I don't have any aspiration that we're going to be able to detect some of these rare plants. Even the relatively uncommon plants are going to be pretty tough to map.
Okay, interesting. Yeah, I'll look forward to talking to you more about that offline. In my experience, some of the more subtle habitats like wetlands, it can be really hard to distinguish between species. Have you had any habitat-specific challenges?
Yeah, and wetlands is one of those. The biggest problem is that they're just undersampled. So they're relatively small environments, and that means that in order to do a good job with wetlands, we need to go out and really do a more thorough job in the field. So wetlands are a challenge. The other thing that's a challenge is some of these like really low cover alpine environments are challenging as well. And the reason that that's a challenge is that these are 1 by 1 meter pixels on the ground, essentially. And in a lot of these alpine environments, there might be only 5% plant cover in a given 1 by 1 meter pixel. And that means that you're picking up mostly the spectrum of the rock and soil in that environment and very little of the spectrum of the vegetation itself. So that's an example of the type of environment where we're probably going to be able to characterize things kind of broadly by geology. But I don't think that we're going to be able to do a good job at distinguishing particular types of alpine vegetation communities.
Yeah, I see what you're saying. Okay, thank you.
Yeah. All right, any other questions about imaging spectroscopy? All right. Yeah, let's move on a little bit. So a lot of my work, especially in the last couple of years, is based on sensors that are mounted on UAVs, on unmanned aerial vehicles. And these are called drones. I want to sort of show you guys a little bit of what the images from drones look like, and also give you a sense of what's possible using drone imagery now, because it's quite a bit more than just taking pretty pictures from the air, although you can do that. So this is an image of Rocky Mountain Biological Lab in May of 2018. And I'll highlight the drone there. This is just a regular sort of consumer drone, a Phantom 4. And what that is doing these really low terrain following flights. So it's flying across the surface of the meadows at a height of about 30 feet. And every couple of seconds, it'll take a picture. And this is what those pictures look like. So the yellow dots that you see are actually individual flowers of, in this case, this is canary violet, which is really common in flower at this time at this site. And this is really cool because what it allows us to do is to sort of see the landscape like a bee does. So right now I'm working with some pollination biologists to see if we can map nectar and pollen for bees using this imagery. And I can show you what the images look like in more detail here. So this is one of the study sites that we've got up a little bit outside of Rocky Mountain Biological Lab. It's about the size of a football field. And I've just had this little red highlight to zoom in on a particular spot on this site. And what you're seeing is a time lapse of how this site developed over the course of the season. So you can see in early May, there's very little that's flowering. And then as you go through June, you start to see the plants grow and develop. And finally, you see a bunch of flowers start to pop out by the mid or late June, including these big cream-colored flowers, which are subalpine buckwheat. It's one of the showiest, largest plants in here. So what we're doing now is we're actually using these images and trying to see if we can get computers to count flowers in images for us. And what that's going to allow us to do is to really get a large perspective, a broad perspective on what's flowering where. And we're hoping to be able to put that together into estimates of how nice a particular meadow is at a particular time for bees and hummingbirds and other pollinators. And also, what things like drought do to that spatial pattern of flowering and the abundance of the resources that they need. So that's going to be pretty fun. We're early in that project, and we're still working out some of the details. But before we go to questions, I did want to actually show you a little bit more about how we process the images to get a map like this. I'm going to go back to kind of industrial strength computer we have going on at Harvard University. That's going to perform a little bit better for what we want to do than my laptop or desktop computer. There should be Zoom should just have hold music. I think that's, hold on. Oh, that does require me to type in my password correctly. Hold on one second. Well, I'm not sure what's going on there, but I can at least show you what the finished products look like in on a map. So I can do that using my other computer, which I was looking at before. So this is one of my study sites that's near Judd Falls. So it's kind of a rocky meadow environment that's, if you've ever been up to Judd Falls, it's off to the left side of the trail as you go up to the falls itself. So this is what's called an orthomosaic. So it's a really high resolution aerial photo that's generated by stitching about 400 individual drone photos together into a map. And we're also doing some processing to remove some of the view angle effects in this map. And this is pretty fantastically detailed. If we wanted to zoom in, you can actually see individual plants and you see little animal trails. You can see the road down here at the lower end of the map. But that's the kind of high flight imagery. Has a resolution of about 3 centimeters. And for a subset of this area that's about the size of a football field, we have also done that really close terrain following flight. So we can see what the imagery, this is essentially the highest resolution drone imagery that we can feasibly collect right now. And it appears that my computer is being a little bit slow, but let's see if we can get this to display. wonder if my computer's decided to install updates or do something else computationally intensive. Oh, there we go. All right, let's see if we can find one of these bookmarks. Aha, there we go. Cool. So this is what the high resolution looks like, image looks like. And so you can see two different patterns of colored dots here. So the yellow there are dandelion, and the purple are larkspur. And we've got this drone mosaic for actually a pretty large area, larger than a football field. And this is equivalent to the area of meadow that, say, a bumblebee would have within its foraging range. And so we're really trying to use this imagery to map flowers across this larger landscape, and then ultimately try and figure out how does a bumblebee navigate a landscape like this, or a hummingbird for that matter. So that's essentially what we're doing with drone imagery. And yeah, I wanted to pause and ask if anybody's got any questions about the drone stuff that we do.
Hi, Ian. Yeah, I wonder if you could tell us a little bit more about how you might classify plants in those images, because there you don't have all of that like spectral detail that you would have in the for classifying trees.
Yeah.
Yeah.
So that's a really important and subtle point. So with the hyperspectral imagery, we had essentially billions or trillions of colors that, and an ability to distinguish between colors that's very very sophisticated. And the trade-off is that we can't fly a camera that's that sensitive 30 feet off of the ground using a drone, at least not yet. And so what we're reduced to is imaging with regular cameras that can only see in red, green, and blue. And if we were just relying on the color information, it would be pretty difficult to distinguish between different types of flowers. But what we can actually rely on is that different flowers have different shapes, sizes, and other geometries. And there's a particular class of algorithms called convolutional neural networks that are really good at learning the particular shape and size of objects like flowers. And so it's using the color information, it's using the size information, and it's also using the geometry and spatial pattern information to decide whether a given blob in the imagery is a larkspur or a columbine or a dandelion. And that right now it works for like the larger plants, and it works in situations where we're trying to distinguish between a few species. But the computer is still a pretty poor botanist. So one of the challenges we're working on right now is how do we scale that up to dozens of different species, some of which look very similar. I really wish that we had a camera that was sensitive outside of just the visible wavelengths, because it might help the classifier. But right now, those cameras are about $40,000, and they're about 5 times too heavy to fly on the class of drones that we're using right now. That being said, this is something that's probably going to be a part of my next grant proposal.
Thanks, very interesting. And so do you use those same approaches for some of the spectral data, or is the resolution so low there that it wouldn't help to disambiguate any of those difficult species?
Yeah, so by spectral data, you mean from the drone images or from the imaging spectroscopy?
Oh, sorry, the imaging spectroscopy.
Yeah, so we haven't tried to observe flowers directly using that information. It was collected in July of 2018. So there should be some areas that, for example, have a lupin in flower that's dense enough to where it probably has a spectral signature. But what that is going to require us to do is, map the spectra of lupin flowers, and then figure out a way of sort of unmixing that from the background, because we can't distinguish individual flowers or even individual plants in the imaging spectroscopy data. It's not fine enough resolution. That's an approach that we haven't really tried, but there are some situations in which you can figure out the fractional composition of different types of surfaces within a pixel, and so that's something that we could try for trying to understand flowers. It's because flowers are pretty low cover in a pixel. Even like the image that I showed you is a pretty dense flower showing, but even then, flowers cover maybe 5% of the surface, and in that case, they're going to be kind of a drop in the bucket spectrally when you look at a coarser scale image.
Yeah, very interesting. Thank you.
Great. So that's essentially all the stories that I had prepared for today's Q&A. But I wanted to leave you guys with one additional point, and this kind of brings back to where I started with this. I started with this idea of this revolution in the ability to see the Earth in different ways using different kinds of cameras, and I think the really exciting thing that I'm working on, and what a lot of my collaborators are working on, is how can we use these new high resolution remote sensing tools to directly link field measurements, like the type of measurements that scientists have been taking at Rocky Mountain Biological Lab for decades, with this new generation of remote sensing tools. And my hope is that we're going to learn a lot more about how these ecosystems work by being able to link these different kinds of data together. And my work is just kind of a tiny little part of that larger effort. But I hope that in the future we're really gonna be able to use images of the Earth at a lot of different scales to better understand our world. And along the way, we're gonna be advancing the mathematical techniques we need. We're going to be advancing the engineering. We're going to be advancing the basic physics. We're going to be advancing the ecology, and it's just an exciting place to be as a researcher. And I really appreciate you guys joining me for this short talk. And with that, I think we'll wrap things up. I will stop sharing my screen. And do you have any closing thoughts before we go?
I have one more question for you, Ian. I'm just curious, how did you end up in this realm of field science? What led you down this career path?
Yeah, thanks. That's a good question. So I've been essentially a nature nerd for most of my life. I would, as a young kid, go out and collect things, and my windowsills and all the surfaces in my house were covered with butterflies and rocks and cool plants that I found, and then I started making maps really young, and sometimes these were fantasy maps of sword and sorcery domains that I had read about. And so when I started to come through school, I was really interested in biology, and I took a course as an undergraduate called Images of the Earth. And that opened up sort of new ways of seeing. And I got really excited about how can we fuse our knowledge of natural history, of our knowledge of how creatures live and grow and make it in this complex world, with those remote observations. And so what I find really really fulfilling is the ability to see the world with a lot of different lenses and use that to learn something new. And that's essentially sent me on a 20-year rabbit hole of remote sensing and ecology. And I did a PhD at the University of Washington. I did a postdoc at Harvard, and I sort of didn't want to sit in my office and sort of be on a more traditional academic track. So I arrived at Rumble because I started doing work here as a postdoc, and then some people got excited about the work that I was doing, and we were able to find a source of funding for it. This is essentially a dream job for me. I get to make maps of nature. I get to have conversations like this, and I essentially just got really, really lucky.
Great. Thank you for sharing with us today. Do we have any final questions? Thank you everyone for attending as well.